So welcome to the lecture on DNA sequencing for MCB 182. And just a reminder, don't distribute the course materials to others outside the class. And so the learning objectives today is really just to understand the advantages and disadvantages of the kind of three main sequencing technologies that a lot of people use today, namely made by Illumina, PacBio, and Nanopore. And have some broad understanding of how they work. And also we want to understand when and why you would use long versus short read sequencing, because long and short read sequencing have their own advantages and disadvantages in general. And so the goal of DNA sequencing at kind of a super basic level is really just to sequence a chunk of DNA, essentially find the complete sequence of bases in a particular sequence. And the challenge to sequencing really is that there basically is no sequencing technology today that can really just take in say a genome sequence of arbitrary length and just spit out its sequence with high accuracy anyways as output in one kind of run. And so typical sequencing technologies, depending on which one you use, can really only sequence say like a couple hundred base pairs or up to say like 10 kb bases at a time. More typically, if you're using like Illumina sequencing machines, you're typically sequencing somewhere on the order of less than a few hundred base pairs. And so as you've probably heard, the cost of sequencing genomes has rapidly decreased over the past few decades. And so just to give you an illustration, this is a graph of the cost of sequencing, say, the human genome. And you can see that back in 2001, when the first draft of the human genome was released, the cost of the whole sequencing project was somewhere on the order of like $100 million. And so that's in contrast to when the first next-gen sequencing machines by Illumina came out in roughly 2007. And you can see at that point, that was when the costs really started rapidly declining. And if you fast forward a little bit to 2015, you can see that was when we first started achieving the so-called $1,000 genome, where sequencing a genome could cost roughly on the order of $1,000. And so these numbers are somewhat misleading because the cost of sequencing a genome kind of depends on how deeply you sequence it. And so if the deeper you sequence a genome, the more it's going to cost, but the more accurate your genome is going to be because you have more and more kind of reads covering any given region of the genome. But this graph really just serves to illustrate that the cost of sequencing overall has just really rapidly declined. And so in terms of the steps for performing, for example, genome sequencing, there's kind of three really broad steps involved. First, you obviously have to prepare a DNA library from a particular organism or cell of interest. Then you need to do sequencing via any number of technologies that we'll talk about later in this lecture. And then the basic output from your sequencing machine is basically a set of one or more reads. They give you a fragment of your original genome or sequence that you're trying to characterize. And so in terms of generating a DNA library, the basic steps, which again kind of depend on the precise technology you're using, but the general steps involve basically extracting some kind of template DNA or genome from a bunch of cells, followed by fragmentation, because as I explained earlier in this lecture, current technologies as of today anyways are not able to sequence an entire genome in general all in one go. And so typically you have to somehow fragment your original genome into smaller pieces and then sequence the individual pieces. And so this fragmentation can be done by any number of ways, including mechanical shearing versus sonication or enzyme digestion. So once you fragment your input DNA sequence into a bunch of fragments, you typically need to add adapters. And so what these adapters are exactly kind of, again, depend on your technology. And so when we start talking about Illumina sequencing, for example, we'll talk about what exactly goes into those adapters. But basically, generally speaking, adapters generally include sequences that help you, for example, do amplification. So they might contain like primer sites, or for example, they might contain complementary sites which allow you to kind of bind these fragments to some kind of like fixed platform. So in Illumina's case, Illumina uses a technology called flow cell, which we'll talk about in a second. And basically the role of the adapters is to help bind and fix these reads to this flow cell so that you can then do sequencing. But once you've added your adapters, basically there's optionally a library amplification step where you might use, for example, PCR to increase the amount of genomic material you have before you do the sequencing. And so generally speaking, when you are constructing your library, there's a number of considerations that you generally have to make. So first of which is obviously the length of your fragments. And so certain technologies like Illumina's sequencing by synthesis technology generally are used for sequencing shorter reads. And so there you might need to do more fragmentation in order to basically construct shorter reads for Illumina sequencing, whereas other technologies like Nanopore can sequence much longer reads, and so you basically need less fragmentation. Another consideration is this library amplification step. So generally speaking, amplification generally introduces errors. So generally speaking, the kind of more involved your protocol is, the more steps there are, then the more chances are that you somehow introduce some errors or bias into your fragments. Because, for example, with PCR amplification, not all reads may get equally amplified. And so when you're trying to decide on your exact library prep, you'd obviously have to decide whether or not you want to do some kind of amplification. And the final consideration is basically the amount of DNA you have. And so certain technologies like Illumina might need a higher number of reads to represent any given fragment in order for you to reliably sequence that fragment. Whereas other technologies that are based on like single molecule sequencing traditionally need less input DNA material. And so this becomes important when you are sequencing certain samples that are rare or are hard to find. So you could imagine that, for example, if you're sequencing cells within a tumor, if you're trying to sequence, for example, so-called cancer stem cells, which are a rare population of cancer cells, you might not be able to get that many cancer stem cells, in which case you might need a protocol or technology that uses less input genome material. And so again, the basic output of any of the sequencing technologies that we talk about today is basically a set of reads. And so what exactly is a sequencing read? A sequencing read is really just a fragment of DNA sequence. So it's basically just a set of bases from one region of the original input DNA sequence. And so between the different technologies that we'll talk about today, there's various considerations that you need to think about in terms of the sequencing reads that you get out of these technologies. So for example, and we'll go into more detail about what the trade-offs are between the different technologies. But basically, when you look across different technologies like Illumina versus PacBio or Nanopore, they generally differ in terms of, for example, runtime. So certain technologies are, they do sequencing faster than others. In terms of cost, generally speaking, technologies that generate longer reads or they can sequence longer reads generally cost more than shorter read technologies like Illumina. A lot of these technologies differ in terms of their error rate, and so obviously sequencing is not a 100% accurate process, and so certain technologies like Illumina sequencing by synthesis, for example, tend to have a lower overall error rate than compared to some of the longer read technologies. Another closely related consideration is that the error rate is sometimes a function of the composition of the sequence. So for example, Illumina technologies tend to be slightly worse at sequencing like GC-rich sequences, whereas other technologies are less biased. Some other considerations are that, again, technologies like Illumina's sequencing by synthesis tend to sequence very short reads compared to like PacBio or Nanopore that can generate like hundreds of thousands of base read pairs or reads. And so why this matters is that obviously, at least in terms of, for example, genome assembly, as we'll see later on in this lecture, because sequencing technologies can only sequence fragments of, for example, the original genome, that means that you have to do additional work in order to kind of piece together your short sequences back into the original genome sequence. And so that process is generally much easier when you're using longer read technologies compared with shorter read. And finally, another consideration is that certain protocols, so most sequencing done nowadays involve what's called paired end sequencing. So what that means is that when you generate these fragments that you want to sequence, sometimes, for example, if you're using short read sequencing technologies, you can only sequence the ends of those fragments and not the whole fragment itself. But most protocols nowadays allow you to do what's called paired end sequencing, which allow you to sequence kind of both ends of each read. And so why that's useful is because if you know approximately what the length of your fragment is, and you sequence both ends, then that means that you have, even though you have two short sequences produced from a single fragment, you know approximately how far they were on the original genome. And so that helps you when you go through the genome assembly process. So the process of stitching back together your genome sequence from your short reads. And so genome assembly is the topic of the next lecture, but just to summarize it in a nutshell, basically the idea is that you start out with some kind of genome sequence, which you're assuming is very long. And then what you're doing is you're going through your library prep process where you're fragmenting the genome, sequencing a bunch of reads. And then so the whole idea here is that when you're doing genome sequencing, you obviously generally don't know what the original genome looks like. So all you have are the short, for example, reads that you've generated from your, say, Illumina sequencing technology. And so genome assembly is the general process of taking those reads and aligning them to each other so that you can kind of figure out which ones are overlapping. And so here, what I'm showing you is the original genome sequence and the reads, and I'm showing you where those reads came from with respect to the original genome sequence. Although you don't know that when you generally doing genome sequencing. You use alignment to figure out which ones follow which other reads. And then in doing so, you can basically reassemble the genome because if you know which reads overlap which other reads, then you can kind of order them from left to right and allow yourself to basically figure out what the original genome sequence was. And so in the process of resequencing for variant identification, as I mentioned in the previous slides, basically the idea here is that somebody's already generated a reference genome for you. For example, for the human genome, you have the draft human genome sequence, which you can download. And the idea here is that if you have a new individual of that species that you're trying to detect, sequence variation of, what you do is you take that new individual, you would collect DNA samples from them, and then you would again generate reads. And you would take those reads and you would map them back to this reference genome. And so the idea here is that because each individual has, sequence variants that are specific to them, when you map these reads back to the reference genome, then for the most part, in the human genome, for example, for the most part, most reads should match more or less exactly back to the reference genome. But when you have positions in the individual's genome that differ from the reference, you're going to have a whole bunch of reads that basically consistently give you a different base at a position compared to the reference genome. And so here you can kind of see that by, if you look closely at the gray boxes, which represent the individual reads, you can see that some of those gray boxes have letters or bases drawn on them, and those are meant to represent sequence variants that are present in the individual you're sequencing, but are different from the reference genome. And so the idea is that, again, here I'm showing what I'm calling is the test genome, is basically the new individual that you're sequencing. And the idea here is that there are certain positions in the test genome that are different from the reference genome, and you can tell that because when you look at the reference genome sequence, so on the top, I'm now showing you some hypothetical bases in the reference genome that differ from the test individual. And you can see that those particular columns of the reference genome, the reference base is different from that of the individual you're sequencing. And so in the variant identification process, basically for each position along the reference genome, you're kind of looking across the reads that you sequenced, and you're basically looking for discordance. And where you see a lot of support, so where there's a lot of reads from your individual that suggest one base that's different from the reference genome, then that's when you can be pretty sure that you've identified a variant specific to that individual that's different from the reference. And so an interesting question to think about is, what would you expect to see for a heterozygous variant, right? So for humans, for example, the human genome is diploid. And so if there's a specific SNP at a position in your genome and your individual is heterozygous in that SNP, then what would your reads look like at that particular site? So here, for example, if we're assuming we have a homozygous variant, then for the rightmost column where the reference genome is an A, and in the test genome I said there was a G, you can see that all of the reads from the individual mapping to that position indicate there is a G at that position in the individual compared to the A in the reference. But the question is, what would you expect to see if you have a heterozygous variant? And so here's just a slide that gives a brief comparison of the three major technologies that we'll talk about in this class. And basically the main differences between them are that Illumina technologies, generally speaking, have a much higher throughput compared to PacBio and Nanopore. And what that means is that generally speaking, they overall can produce a larger amount of sequencing reads compared to the other two technologies. Illumina also tends to have higher overall accuracy, so lower error rate. We'll talk later about PacBio sequencing, but you can achieve a higher accuracy in PacBio because essentially for any given fragment, you can actually sequence it multiple times, which would obviously decrease your error rate over time. But basically, if you were to only sequence each fragment once, then Illumina sequencing generally yields higher accuracy. In terms of bias, though, it turns out that Illumina technologies, generally speaking, are more biased compared to PacBio and Nanopore. What that means is that, for example, Illumina error rates tend to be much higher for GC-rich sequences, for example, whereas PacBio and Nanopore are generally less. The error rate is generally more uniform across different types of input sequence. Generally speaking, again, Illumina it tends to be cheaper on a per base level for sequencing compared to PacBio or Nanopore. Where this trade-off comes, though, is that Illumina technologies tend to be, are used for generating short reads. So they're only really able to sequence reads on the order of a couple hundred base pairs, whereas PacBio and Nanopore can effectively sequence much longer reads in the hundreds of kilobases long range, and nowadays even potentially into the megabase long reads. And so generally speaking, you would essentially select Illumina if short reads are okay for your particular application, whereas if you need long read technologies, then you basically have to go with PacBio or Nanopore. And so there are, the sequence technologies that people use nowadays, again, kind of come down to Illumina, Nanopore, and PacBio, but they weren't really the first sequencing technologies available. And so the Sanger method was really kind of the first generation of DNA sequencing methods that were used, and it was basically invented by Fred Sanger back in the 70s. And so this method was initially used to basically sequence shorter genomes. Interestingly, Fred Sanger is one of the few people who actually won 2 different Nobel Prizes. So that's an interesting tidbit. So DNA polymerase plays a pretty central role in most DNA sequencing technologies. And so just as a brief reminder, DNA polymerase can take a single-stranded DNA template, and after that second strand is being primed, DNA polymerase basically incorporates additional nucleotides in the iterative fashion to elongate that initial second strand in the 5' to 3' direction until it eventually, in theory anyways, can completely generate a double-stranded piece of DNA in a mostly error-free way. So the classic Sanger sequencing protocol essentially needs a single-stranded DNA template, a primer that you can anneal to the start of the template strand, DNA polymerase, and very essentially you need both standard deoxynucleotides, so that's represented by the DNTPs, and you also need fluorescently labeled dideoxynucleotides, or what's represented as the DDNTPs. And so the point here is that DNA polymerase, after the annealing of the primer happens, will start elongating that initial primer using the standard deoxynucleotides. But as soon as one of the dideoxynucleotides gets incorporated, elongation will actually stop because the dideoxynucleotides lack a 3' hydroxyl group that you typically need to chain 2 nucleotides together. And so the key point here is that when you add in the dideoxynucleotides, you add them in at a much lower concentration than you add in the standard deoxynucleotides. And so what typically ends up happening is that, the DNA polymerase will elongate the chain, and only infrequently will you actually incorporate a modified base and stop elongation. And so the idea is that in this Sanger sequencing setup, you have 4 different chambers where in each chamber you only add one of the dideoxynucleotides. And so you're only in each kind of chamber, you're only stopping the chain at one specific kind of predetermined base. And so what you can do is that after your multiple rounds of elongation, basically you end up with a whole collection of fragments of different lengths. And so these double-stranded DNA fragments are denatured, and then you're basically size sorting them using gel electrophoresis. And so what you're going to end up with is a gel that looks like the gel shown on the bottom right here, where basically in each kind of lane you have a lane corresponding to a chamber where you had only one modified dideoxynucleotide base. And so you're going to end up with, when you have a high enough concentration of molecules, you're going to end up with a whole bunch of bands where the elongation stopped. And so the theory is that you can start, basically the shortest sequences will obviously travel the farthest on the gel, and so you can essentially read your DNA sequence by reading the gel from the bottom to the top and looking at, for each row, which letter happens to, or which chamber happened to produce a sequence of that particular length. And so I mentioned earlier that generally speaking, you can't sequence a whole genome all in one go. And it should be pretty obvious from the previous slide why that is. So, if your genome sequence is like, say, 3 billion nucleotides long, it's not like you can run a gel that has a few billion bands on it. And modern day Sanger sequencing doesn't use gel electrophoresis. They do Sanger sequencing within sort of microwell plates. But even then, it's basically impossible to Sanger sequence an entire genome all in one go. And so the general principle that genome sequencing is based on is something called shotgun sequencing. And so again, the idea is that if you start out with, say, a really long genomic fragment or a segment of a genome, the idea is that you cut that genome segment into many different pieces. And so because you obviously have more than one copy of the genome in hand, you cut many times across the genome sequence at random. And so that's what the term shotgun is referring to. And the idea is that for each one of these little small pieces, you can do, for example, paired end sequencing. So we'll talk about paired end sequencing with Illumina later. But basically, for each smaller fragment, you can generate one or two reads from either end of these smaller fragments. And then once you sequence these smaller fragments, the idea is that you can piece them back together and get the original genomic segment. And so in practice, there's two ways that shotgun sequencing is generally used to sequence genomes. So one is called whole genome shotgun sequencing, and the other is hierarchical shotgun sequencing. And so what whole genome shotgun sequencing refers to is you can actually take, for example, an entire genome sequence like the human genome, and you can actually just straight out fragment the entire genome into small little pieces, sequence either one end or two ends of each small little fragment, and try to assemble the entire original genome sequence from these very small fragments. On the other hand, an older style approach is what's called the hierarchical shotgun sequencing approach. And so that approach is a little bit more structured in the sense that you take your original genome sequence and you first divide it into larger fragments, like what are called bacterial artificial chromosomes. And these larger fragments tend to be on the order of, say, like 150 kb to 350 kb. And then for each individual segment that's 150 to 350 kb long, you then shotgun sequence that, and then try to basically sequence short little fragments, reassemble the individual backs, and then somehow put the backs back together. And so it's called hierarchical because you are kind of dividing the huge genome into smaller pieces, and then finally sequencing the smaller pieces and kind of building it back up. And so there's a kind of long history and there was a lot of controversy when the human genome was first sequenced, because there were kind of competing camps. There was one camp of people who thought that hierarchical shotgun sequencing was the way to go because it's very structured in the sense that you're dividing the genome into smaller and smaller, kind of more manageable parts that you sequence before you try to build it back up. And then kind of the so-called newer camp were the people who thought, this hierarchical approach is really slow because, you have to organize these different back libraries and piece them back in a very hierarchical fashion. And so they thought that, based on computer simulations, they thought that you could actually, you could just throw away this whole idea of having to build this hierarchical back system, and you could just fragment the original genome into these small pieces and just somehow use computers to assemble them back to the original genome sequence. And so whole genome shotgun sequencing is basically the approach that they kind of won out in the end, and that's kind of more standard nowadays compared to hierarchical shotgun sequencing. But it is kind of interesting to note kind of these two major different approaches to genome sequencing. And so going back to this idea of these backs, so basically, again, a bacterial artificial chromosome, or back, is basically just like a synthetic DNA construct that you use to clone DNA sequences into, say, for example, like bacterial cells like E. coli. And so the idea is that if you're trying to sequence, say, a human genome, what you can do is you can take a whole bunch of human cells, you isolate the genome sequence from those cells, and then you basically break them up into large 150 to 350 kb segments. And then for each one of those segments, you can put them into some vectors, transform them into, say, for example, like E. coli, and then basically generate your library from that. And so the idea again is that each individual back contains one large fragment from the original genome. And then because you have multiple copies of the original genome sequence, then you can have backs with kind of like overlapping regions of the original genome. And so what's kind of an interesting point to make is that back when the human genome was being sequenced, The initial draft of the human genome was actually generated based on backs from just a handful of anonymous volunteers. And actually about 3/4 of the initial draft sequence actually just came from 2 back libraries. And so I mentioned earlier that in terms of hierarchical shotgun sequencing, you have to kind of sequence individual backs, and then you kind of have to piece these backs back together. And so, the process of piecing the backs back together is not entirely straightforward. But the idea is that, assume for the moment that you've somehow managed to sequence individual backs, right? And so the idea is that in order to kind of figure out how to piece individual backs back together into the original genome, you basically need to build what's called a physical map of the backs. And so just as a quick review, a physical map of a genome means that you're basically trying to build a map of the genome where you know the absolute position of different DNA sequence elements. And so what that means is that you know the rough or precise position of where different restriction sites are, or transcription start sites in the genome. Like the transcription start site of like GATA4 is at like, position 11 billion of chromosome 5 or whatever. And that's kind of in contrast to, again, what is generally referred to as a genetic map, where genetic maps are basically maps derived from knowing the relative distances between genetic landmarks. And so by relative distance, I mean that through other means, you can generally estimate, for example, the relative frequency of crossover events that occur between 2 different genetic landmarks on the genome. And so basically by estimating the frequency of crossover events, you can generally speaking determine which, how far apart two different genetic landmarks are. And so for the purposes of assembling backs back into the original genome, it's the physical maps that you really need to know about. And so the idea is that, suppose you took, for example, a single back, right? And so what you could then do is you could take a restriction enzyme, or say a collection of restriction enzymes, and you can use those restriction enzymes to cut at different restriction sites along a single back. And so the general idea is that if your restriction enzymes have relatively high sequence specificity, which they often do, and your back is long enough, what that means is that any given fragment of the original genome, which corresponds to an insert in a back, will generate kind of a unique banding pattern on a gel. Because, if your restriction enzymes are highly sequence specific and your, genome is of high sequence complexity, then you're going to get a unique, set of fragments after restriction enzyme cutting. And so here, suppose that numbers 1, 2, and 3 refer to, the inserted genomic fragments in 3 different backs. And suppose you, in this hypothetical case, you got lucky and, insert 1, 2, and 3 are overlapping. And so the idea here is that if you use the same set of restriction enzymes to make cuts along fragments 1, 2, and 3, then if you look at fragments 1 and 2, for example, because they overlap, the restriction enzymes will basically make the same cuts along that overlapping fragment. And so what that means is that 2 inserts that share sequence will have corresponding fragments after the restriction enzyme cutting of the same length and sequence. And so that means that you'll have overlapping bands in your gels between insert 1 and insert 2. And similarly, because insert 2 and insert 3 have overlapping sequence, after restriction enzyme cutting, they're going to share a band, at least one band, because because they have the same sequence in that overlapping region. And so after restriction enzyme cutting, that'll yield an overlapping band. And so the general idea is that by finding common bands across multiple backs, you can kind of get an idea of which backs are overlapping each other on the original genome sequence. And that will then kind of allow you to kind of assemble those backs and figure out which ones follow which. And so the big picture here is that, if you start with the original genome sequence and you fragment that genome into many different pieces and generate your back library based on, creating these 150 to 350 kb inserts and putting them into the backs. The idea here is that through physical mapping, you can basically figure out where, broadly speaking, each insert came from with respect to the original genome. And because you started out with many copies of the original genome sequence, your back library is going to be highly redundant, right? What that means is that you're going to have, if you consider all of the backs in your back library and where the insert came from on the original genome sequence, you're going to have a lot of different regions being represented by multiple backs. And so the idea then is that in order to do hierarchical genome sequencing or hierarchical shotgun sequencing, you then want to take your original highly redundant back library and you just want to select a subset of backs that more or less are not that overlapping with respect to the original genome. Because the less overlapping each back is, then that basically means the less wasted sequencing happens, right? Because if you're sequencing a whole bunch of backs that are, have a lot of overlap, that means that you're kind of wasting money because you're just sequencing the same thing over and over. So you want to make sure that the backs that you actually sequence are, generally speaking, not that overlapping. And so, since Sanger sequencing was really developed, a number of technologies have now been developed which have higher throughput than Sanger sequencing. And so the three main ones that we'll talk about in this lecture are sequencing by synthesis, which is kind of spearheaded by the Illumina sequencing technologies. You have the PacBio smart cells, and you have Nanopore sequencing. So next we'll talk about Illumina sequencing by synthesis, or SBS technology. So it's worth pointing out that Illumina has released a number of different products that are all based on the sequencing by synthesis technology. And so the SBS technology that we'll talk about in the next few slides applies to all of these different products. But it is worth noting that there are different products that are designed for different, for example, throughputs. And so if you are doing a lot of just kind of small scale sequencing experiments, you might choose a technology like the MiSeq. Whereas if you're doing a lot of really large scale, say like human genetic association studies, then you'd probably use something like the HiSeq X series. And so the basic strategy for doing sequencing with Illumina SBS is just like, as we saw before, you start out with a genome sequence, and you have to fragment that sequence into small pieces. And what happens is that for each individual fragment with Illumina SBS, you are basically sequencing both the 5' and the 3' end of each read. And because Illumina's technology is based on short read sequencing, each read that you generate from a given fragment is really only on the order of 75 to 300 base pairs long. Generally speaking, because you know the rough size of the each fragment, the actual information that you get out of Illumina sequencing is that you know the length of the reads on either end of each fragment, and you also know the approximate distance between those two reads, and so you get a little bit more information than just the read sequence itself. And so in terms of DNA library prep for Illumina, the basic protocols basically follow the general outline that I gave in earlier slides. So generally speaking, you start with your genomic sequence, you do the fragmentation, and before you do the sequencing, you basically just need to ligate some adapters. And so on the next slide, we'll basically talk about what exactly those adapter sequences do. But what it is worth pointing out is that as part of these adapter sequences, you can typically add what are called sample indices or barcodes, which basically allow you to multiplex multiple samples together to be sequenced. And so the main point here is that one of the reasons why sequencing costs are dropping so quickly is that oftentimes when you generate fragments to sequence from a given sample, the amount of fragments that you're sequencing for any given individual sample is actually relatively low compared to the total number of or total amount of DNA sequence you could sequence in a run of one of the Illumina technologies, for example. And so if you sequence a sample that is much smaller than the total amount of sequencing that you can actually generate from a run, then you're essentially wasting capacity because you could sequence more. You just didn't have enough fragments from one sample. And so sample barcoding is a way of basically grouping together multiple samples and sequencing them all at once on the same run. But the point of these barcodes, again, is just to allow you to figure out which reads came from which original samples. So this slide illustrates the basic structure of the Illumina adapters. And so it's worth pointing out that there are multiple different types of Illumina adapters, depending on the specific protocol. But there's a few features that are pretty common across all of them. And so the idea is that in the dark gray box basically represents your fragment that you're trying to sequence. And so in terms of the adapters on either side of the insert, you have, for example, what are called the P5 and P7 binding sequences. And so as you'll see in the next few slides, the P5 and P7 sequences are generally just there to allow you to bind your fragments to the flow cell or to the sequencing instrument in order to do the actual sequencing. On top of that, you have what are called the sample indices. And so in the single index case, you have what's called the I7 index. And so this I7 index is basically what allows you to multiplex multiple samples together for sequencing. So if you were to sequence, say, for example, 3 samples all in the same run of Illumina, each sample would basically get adapters with its own unique I7 index, so that when you do the sequencing, you know which read came from which sample. Finally, you have the SP1 and SP2 sequence primer sites. And so these are basically just primer sites that allow you to do, for example, like amplification or actual sequencing. And they basically flank the actual insert sequence. And so those sequence elements are common across all of the Illumina adapters. So the difference between a single index and a unique dual index adapter for Illumina is that what people noticed is that, so in theory, your sample index, your I7 index, is all you would need to identify which sample your read came from. But the problem that people noticed after using the single index adapters after a while is that they started noticing that when you multiplex multiple samples together, you actually end up getting what's called index hopping. And so what that means is that you can actually get incorrect I7 or you can get incorrect I7 indices that don't match the correct sample that they came from. And this is largely the result of free adapters that did not get ligated to specific fragments. And so the important part here is basically that in the dual index adapter, you basically have an additional sample index of what's called an I5 index, which is basically just another sample index that allows you to, again, kind of uniquely identify which fragment belongs to which sample. And so the idea is that when you add these I5 and I7 indices to each sample during your adapter ligation, you're giving a unique I5 and I7 pair to each sample so that each one uniquely identifies each sample. And that just basically allows you to make sure that even if index hopping happens, you can kind of identify it because you'll have mismatched I5 and I7 indices. Finally, there's what's called the dual index UMI, which basically just means that one of the adapters has an additional what's called a UMI or unique molecular identifier tag associated with it. And that just really allows you to identify unique reads. And so basically, because each fragment has its own UMI molecule or its own UMI tag, that means that even after amplification, you can distinguish sequencing two different essentially PCR duplicates, because when you have a PCR duplicate, you'll have two sequence fragments with the exact same UMI. And so central to Illumina sequencing technology is what's called the flow cell. And so the flow cell is basically just a glass slide that has different lanes imprinted on it. And the idea is that within each lane, you have a set of oligos that are basically covalently attached to the glass slide. And these oligos have complementary P5 and P7 sequences on them. And so the idea is that when sequencing actually occurs, what happens is that you're passing your DNA fragments that have the adapters ligated to them over each lane. And because your adapters have those P5 and P7 indices on them, they'll basically hybridize to the complementary P5 and P7 oligos that are fixed on the slide. And what's going to happen is that your individual DNA fragments are going to randomly hybridize and get fixed across the entire lane. And then basically, once they're fixed onto the glass slide, then sequencing occurs. So the idea of the sequencing by synthesis technology is that once you've done your library prep and you've added your adapters to your DNA fragments, what you then do is you take your fragments and you pass them over the flow cell and allow the fragments to hybridize to the P5 and P7 complement adapters that are fixed on top of the flow cell. And so after hybridization, what you end up with is a whole series of DNA fragments that are spread out all throughout the lane, hybridized to different regions along the lane. And then you basically perform a process called bridge amplification. And so the idea of bridge amplification is that each of your individual fragments from your library prep is kind of spread out throughout each lane of the flow cell. And bridge amplification really just tries to increase the number of copies of each fragment that are kind of located at a given position along the lane in the flow cell. And so the idea is that ultimately the way sequencing happens is that after bridge amplification, you actually use DNA polymerase to synthesize additional copies of your fragment. And as you're doing this synthesis, you're actually essentially taking pictures of the bases as they're getting added during synthesis, and in doing so, you're sequencing by imaging essentially. And so you need bridge amplification in order to increase the number of copies of a given fragment at a position in the flow cell in order to generate signal for your pictures. And so in the next few slides, I'll actually go into bridge amplification and the actual sequencing by synthesis more. But for the time being, just, for the purposes of this slide to show you the overall process, just know that you need to do bridge amplification in order to increase the number of copies of a fragment on a position in the flow cell. And so once you've completed bridge amplification, that's when you actually do the sequencing by synthesis. And so this slide is basically here just to show you in slightly more detail what bridge amplification involves. And so again, you start out with your double-stranded genomic fragment with the P5 and P7 adapters attached to the end of it. Then you can see that because it's double-stranded, basically there's a P5 and P5 complement as well as a P7 and P7 complement. And so on the other hand, on the surface of your flow cell, you have both the P5 and the P7 primers that are attached on the surface. And so the idea is that when you denature your double-stranded DNA molecule, basically the complement of the adapters can bind to the P5 or the P7 primer that's attached to the surface of the flow cell. And then through a process of extension and then basically denaturing, followed by some binding and extending, you can basically turn like one genomic fragment that's bound to the surface of the cell into two, and then two can become four, and then four become eight, and so on and so on. And so basically at the end of the day, the diagram on the left basically shows you that without bridge amplification, basically your lawn has many P5 or P7 adapters fixed on the surface of the flow cell, and your genomic fragments are kind of sparsely bound across the surface of this flow cell. But after bridge amplification, basically you end up with a whole bunch of copies of the same fragment in spots across the flow cell.
So after bridge amplification finishes, the next step is the actual sequencing by synthesis. So at a high level, after you add a sequencing primer template, sequencing by synthesis involves basically sequentially extending the template one base at a time by providing DNA polymerase with 4 different modified nucleotides. And so the key idea here is that these modified nucleotides have 2 additional components, a fluorophore and a reversible terminator. And so the idea is that when you have a template and you start adding these modified bases, DNA polymerase is going to pick up the appropriate complementary modified base, and it's going to extend the template by 1 base. And the terminator that's sitting there is going to basically prevent further elongation of the template. And so basically what will happen is that exactly 1 modified nucleotide is going to get added. And so because of the fluorophore, basically that additional base is going to emit a certain color. And so that color can be imaged and recorded so that you know which base was incorporated at which position of the flow cell. And so after the imaging happens, then basically you can cleave the fluorophore, you can remove the terminator, and then you can have another round of synthesis where another different modified nucleotide gets incorporated. So on the previous slide, I basically showed how sequencing of a single fragment can occur from one end. But previously we talked about how it's pretty common actually to do paired end sequencing, where you basically get reads from both ends of a given fragment. And so this slide is basically an illustration of how paired end sequencing works. And so steps 1 through 3 are basically pretty similar to the previous slide, where you basically start with some fragments with the adapters ligated to them, and you do some bridge amplification to generate clusters of fragments representing the same sequence at a given spot on the flow cell. And so in step 4, what happens is that you actually cleave all of the fragments that are bound by the P5 adapters specifically. And what happens then is that that gives you clusters that only has fragments that are bound by the P7 adapter. And so that really makes sure that all of the fragments that you sequence in a cluster are all in the same direction. And so the sequencing primer then basically anneals to the P5 end of the fragment. And then you start the sequencing by synthesis step that we talked about in the previous slide. And so here read 1 refers to basically the freshly synthesized strand. And so once you are done sequencing one end of the fragment, basically in step 5, what you then do is you sequence the sample index I7 in those cases where you actually barcoded your samples. And so the indexed reads are basically, so just to emphasize there, the index reads are only used when a sample is barcoded. And so when read 1 is finished, basically everything from read 1 is removed and an index primer for I7 is added. And basically that will anneal to the P7 end of the fragment. And then you end up sequencing the index sequence. And so in step 6, basically what you do is you do another round of bridge amplification, and that'll regenerate fragments that are also bound by the P5 adapter. And then in step 7, this time you actually cut the fragments that are bound by the P7 adapter instead of the P5. And so that basically results in clusters that only have fragments that are attached by the P5 region. And then finally in step 8, you basically add another sequencing primer for the P7 region. and then you sequence basically what amounts to read 2, which is the other end of the fragment.
And so here's a hypothetical example of a series of images that we might get by doing sequencing by synthesis. And so the idea is that each black square here represents a picture of the flow cell, the lawn on the flow cell. And so you basically see colored dots wherever there was at least one strand bound to the surface of the flow cell. And so as you move through multiple cycles of synthesis, you basically end up with different sequences of colors on each spot corresponding to different sequences that are being synthesized on each spot. And so in practice, you're not manually looking at these pictures and trying to, write down the set of colors that you see on each spot. You use software called base colors, which basically takes a series of images that are generated through each cycle of synthesis. And this software is basically, it basically identifies the spots that correspond to genomic fragments on the lawn. And the software basically just reads off the colors that it's seeing after each cycle of synthesis. And then gives you basically a file back to you that contains, the sequences of all the reads that it saw corresponding to the different spots on the lawn. And so sequencing by synthesis is mainly limited to, you can really only generate reads of length, say like 100 to 200 base pairs. And one of the reasons for this is because templates essentially get out of sync. And so, as I mentioned previously, through bridge amplification, you end up with these spots on these flow cells where each spot contains like a dense set of fragments that are bound to the flow cell. And so in theory, with each cycle of sequencing by synthesis, you're only adding like one nucleotide to each primer that's being elongated. And so in theory, after say like 10 cycles, each strand should have the primer plus 10 added nucleotides. But the problem is that you can get out of sync because sometimes you'll incorporate no extra nucleotide on a given strand, or sometimes you'll incorporate like 2 or more nucleotides. And so after like say 100 cycles, the chances of strands being in different cycles effectively is pretty high. And so that's why your effective read length can only be about 100 to 200 base pairs. Base callers can kind of detect when this kind of out of sync problem occurs, because basically at the beginning in the early cycles when everyone is in sync, the colors are very definitive and it's easy to tell what's red and what's green and so on. But as you get more and more out of sync, the colors start mixing, and so you get less and less pure signal. And so the base callers are able to look at those images and see kind of like distortions in the color, and then estimate some level of base quality for each base in the read that it sequences. So sequencing machines spit out what's called FASTQ files in order to give you your sequences back. And so in a FASTQ file, which I've shown an example of at the bottom of the slide, you basically have sets of 4 lines where each set of 4 lines corresponds to one read that was sequenced by the machine. And so line number 1 basically just represents the name of the read. Line 2 is the actual sequence that was produced or called by the base caller. Line 3 is just a placeholder that we'll ignore here. But line 4 is actually the line that corresponds to the quality values that are estimated for each base call. And so I was mentioning earlier that the base callers try to give you like a quality estimate to tell you how good it thinks the called base is at a given position in the read. And so you can see that the quality value sequence is a series of like letters like H, G, I, dashes and numbers, and those characters basically correspond to different quality values that are estimated by the base caller. So I want to briefly discuss the notion of sequencing bias in the Illumina technology. And so generally speaking, reads generated by sequencing by synthesis basically lead to poor representation of GC-rich and GC-poor sequences. And so the reason this is a problem is that, of course, if your genome sequence has regions that have either very high or very low GC content, then those regions are going to be poorly represented in the reads that you get after a sequencing run. And so obviously with poorer coverage, that means that it's going to be harder to, for example, assemble those regions of the genome because you don't have reads corresponding to them. This also has an additional slight problem in that if you have poorer coverage of the GC poor and GC rich regions, that means that the regions that are sequenced might tend to get higher coverage, relatively speaking, in your data, which then might tend to lead to those regions being identified as repetitive or at least overrepresented in your data, which can also be a problem. And so there's two main sources of sequencing bias in Illumina that people have looked at. And so the main source of sequencing bias is basically PCR amplification. And so more specifically, it's PCR amplification that's done in the library prep after you ligate the adapters to the ends of your genomic fragments. And so the PCR amplification bias, generally speaking, comes from, at least in the case of the Illumina library prep protocol, is driven by incomplete denaturation of, for example, GC-rich regions. And so to fix this problem, there are, for example, PCR-free ways of doing the library prep now. But generally speaking, they need a lot of input DNA as compared to the protocols that use PCR, obviously. And so there isn't a perfect solution to this problem. And the other source of bias comes from bridge amplification, namely that, for example, in bridge amplification, you also have a denaturation step. And so depending on the GC content of your strands, that can also introduce bias.
So I want to briefly discuss the concept of library complexity. And so the idea behind library complexity is that when you sequence a library, depending on how deeply you sequence it, you can oftentimes find that there are sequences or reads that are duplicates of each other. And the reason for this is that suppose, for example, that you obtained a sample and you didn't really have enough DNA to do sequencing on it. What oftentimes happens is that you might do a few extra rounds of PCR amplification and just to get enough input DNA material to put into the sequencer. And so the problem you have is that if your starting genomic material, for example, was too small, or you over-sequence your library, what's going to happen is that you're going to end up sequencing a lot of PCR duplicates. And so PCR duplicates will, in the absence of sequencing error, will look like identical reads, sequence identical reads. And so this is bad because if all you're doing is sequencing duplicates, then that's not really giving you additional information, and you're really just kind of wasting money doing extra sequencing that you didn't have to do. And so a quantitative metric of library complexity is what's known as the non-redundant fraction. And so if you take, for example, if you have like 100 reads that you've sequenced from a library, the idea is basically that of those 100 reads that you sequence, you can count the number of unique reads. So the number of reads you have after you remove any duplicates, and you take that number and you divide it by your total number of reads that you sequenced, and that's your non-redundant fraction. And so the idea here is that if every read is unique, so you haven't sequenced any duplicate, then your non-redundant fraction will be 1, and that means that you have high library complexity. On the other hand, for example, if every read has 1 duplicate, then what you'll find is that your non-redundant fraction is 50%, or 0.5, which means that it's low. And so the NRF is basically a nice kind of easy statistic you can calculate that gives you some idea of how many PCR duplicates are you sequencing, essentially.
So in terms of throughput, depending on which Illumina sequencing machine you purchase, for example, for the Illumina X10, you can produce something on the order of like 1.8 terabases in less than like 3 days. Your reads, generally speaking, are limited to, again, somewhere on the order of like 150 base pairs. And you can do like paired end if you want. And so basically the main advantages of the Illumina approach, the sequencing by synthesis approach, is that you, in terms of raw throughput of the three technologies we'll talk about today, you basically get the highest throughput with Illumina. Accuracy is generally speaking pretty high, and it's pretty easy to do. The main disadvantage of Illumina is that your read lengths are really short, and so from a practical standpoint, you get about 150 base pair reads, and this becomes a problem for sequencing, for example, like repetitive, genomes with a lot of repetitive sequence, or genomes with a lot of like duplication, and we'll see why later.
So short read sequencing is great in large part because it's cheap, fast, and high throughput, but it does have its limitations. And so first of all, detecting structural variation in a genome is pretty difficult with short read sequencing. And so I'll discuss exactly why this is in the next few slides. But basically, in short, when you have, say, for example, a reference genome, and you're sequencing a new genome, and you want to detect events like duplications or inversions, you typically need reads that span the boundaries of, for example, where the inversion happened in order to actually detect it. And so having reads that span these kind of boundaries is easier when you have longer reads. And that'll become more clear in the next few slides. And so for reasons including detection of things like structural variation, and also for, due to problems of trying to sequence repetitive regions using short read sequencing, which we'll discuss in more detail in the next lecture, actually. Doing tasks like sequencing genomes de novo is really difficult with short read sequencing, and so long read has a major advantage for that particular task. Another thing that I want to briefly mention is the issue of sequencing bias. And so when we talked about Illumina SBS technology in the previous video segment, I mentioned that both bridge amplification and PCR amplification introduce bias into the sequences that you generate. And so PCR can actually be used in long read protocols as well. But bridge amplification is a step that's specific to Illumina technology. And so by switching to, for example, long read sequencing, you can at least eliminate bridge amplification as a source of bias.
So one of the main benefits of both PacBio and Nanopore over Illumina is that they give you much longer reads. You can easily get, say, for example, like 10 kb reads compared to Illumina's 150 base pair reads. And so this helps you out in a number of different scenarios. And so, for example, suppose that you're interested in studying structural variation in the human genome. And so, for example, if you're interested in sequencing regions that have segmental duplications, so segmental duplication is basically a duplication of a relatively large genomic region, say like at least 1 kb long. Part of the problem is if you have short reads, in order to distinguish the fact that your genome has, say, 2 copies of a given, in this case, blue genomic region, you need to have reads that span both the duplicated sequence, so the blue, one of the blue arrows, as well as the flanking sequence, and that flanking sequence has to be different across your 2 copies. And so with short reads, typically that's basically impossible. And so when you do short read sequencing of duplicated regions, oftentimes you'll end up with what's called a collapse in assembly. So instead of seeing 2 different copies of a given region, you'll end up seeing just 1 copy in your final assembly because you didn't have enough reads that span both the duplicated region as well as the unique flanking sequence of that region. And so obviously with long reads, if you have long reads, your chances of getting reads that span both the duplicated region and the unique flanking sequence is much higher. And so you'll tend to be able to resolve these kind of duplications. And so similarly, if you're looking at events like inversions, To be able to detect an inversion in one individual's genome versus, say, a reference, you need to have reads that span the region that's being inverted as well as the flanking sequence. And so, again, that's pretty hard with short read sequencing, and so you might not detect that inversion event, but you'll be more likely to detect it with longer reads. And so longer reads have major applications in, for example, helping to assemble genomes. Another area where long reads help us is basically in doing what's called haplotype phasing. And so, in short, we'll get to this, we'll go into this in more detail in the genetics lectures. But basically, obviously, between, say, for example, different humans in the same population, you oftentimes see, you can see a lot of common variants or SNPs that are different between pairs of individuals. And so one of the goals of human genetics is to be able to accurately sequence, all of the humans on the planet. And so what that involves is not just determining for each particular SNP in the genome what is each individual's genotype at that position, but another important goal is to be able to phase that, do haplotype phasing. And so what that means is you want to be able to determine which variants sit on the same chromosome within a given individual. And so haplotype phasing is very hard with short read sequencing, because if your reads are only a few hundred base pairs long, then the chances of sequencing two different SNPs on the same read is generally pretty unlikely. But if you have long read sequencing, then this becomes much easier, obviously. So this slide basically just shows you that when you have long read sequencing, then your reads will tend to capture multiple SNPs on the same read, which then allows you to phase the genome because you know which SNPs occurred on the same chromosome. So the first of the two technologies we'll talk about is PacBio sequencing. So for PacBio sequencing, your average read length is on the order of 10 to 15 kb long, although you can actually theoretically go up to about 100 kb, which is obviously much longer than Illumina's 150 base pairs. In terms of accuracy, the accuracy is around, it depends on what kind of sequence you're sequencing exactly, but your accuracy sits somewhere on the order of like 88% or more. And this compares to Illumina, which is, more than 95%. The throughput obviously is on average much less than Illumina. So similar to the Illumina sequencing protocol, in the PacBio library prep step, you're taking genomic DNA, you're fragmenting it into smaller fragments, and then you're adding some adapter sequences on the ends to help with sequencing. Unlike Illumina, the adapter sequences are actually hairpin loops. And so in what's called the SmartBell template, basically you're adding hairpin loops to either end of a genomic fragment to form these SmartBell templates. And so the interesting thing about the SmartBell template is that they're essentially structurally linear, in that it's mostly basically just a linear strand of double-stranded DNA with some hairpin loops on the ends. But it's topologically circular in the sense that if you treat it as a single-stranded piece of DNA, then it's basically a circular piece of DNA. And so these hairpin adapters are somewhat special in the sense that they provide a common primer binding site for DNA polymerase. And so in the actual PacBio smart cell that we'll discuss in the next slide, you can attach the SmartBell template to, or you can bind it to primers specific to SmartBell adapters, as well as DNA polymerase to facilitate sequencing. So in the PacBio smart cell, a smart cell is basically made up of thousands of small little wells, where each well is like a little microscope with a light source at the bottom of the well. And so within each well, you have the light source on the bottom, and you also have the SmartBell template with one DNA template, as well as DNA polymerase fixed at the bottom. And so the idea is that once you add the primer that gets annealed to the hairpin loop, you start adding nucleotides that are again labeled with different colored fluorophores. And as each base is being incorporated into the template, you basically send a pulse of light and a color is emitted depending on which base gets incorporated. And so kind of similar to the Illumina case, you're basically observing sequences of colors, which you then kind of translate into an actual DNA sequence using a base color. And so some of the interesting aspects of the PacBio technology are that, number one, there's no, in some sense, theoretical limit to the length of the DNA fragment you can sequence, even though I think in practice you can't really go beyond 100 kb. Another interesting aspect is that even though the error rate is higher than Illumina, the errors are more random. And so what that means is that there's less bias in the sequencing, in the sequences that you get. So for some types of sequencing technologies, GC-rich sequences, for example, tend to get more errors in them, whereas with PacBio, the errors are kind of more evenly distributed among the different types of DNA sequences that you can sequence. And so another interesting aspect of the PacBio system is that because the SmartBell templates are circular, what that means is that each SmartBell template can get sequenced multiple times in theory. And so here's basically a visualization showing you that after primer is annealed to the template and DNA polymerase extends that primer, by the time it goes all the way around the entire circular piece of DNA, it can kick out the old template and just keep sequencing over and over and over again the same template. And so that provides benefits because if you have like a small DNA fragment, that allows you to sequence the same fragment multiple times over. And so then you can correct for errors, because if you sequence the same piece of fragment over and over and over again, then if the errors are really random, then you can just take the consensus sequence from the fragments that you did sequence, and then you'll get a more accurate consensus read from that. And so the final technology we'll talk about in this lecture is basically nanopore sequencing. And so with nanopore sequencing, your total, your maximal read length is actually over like a megabase, which is an insanely long read. The accuracy is still relatively high. It's somewhere on the order of 78 to 90%, depending on what kind of sequence you're looking at. And the throughput again is relatively low compared to Illumina, but still you get these super long reads. So at the heart of nanopore sequencing are proteins called nanopores, which are basically pore-forming proteins that are canonically used to transport molecules across, for example, cell membranes, and also DNA helicases, which, as you know, can unwind, they're enzymes that can unwind double-stranded DNA. And so in the nanopore sequencing setup, there's basically a synthetic membrane with these nanopore helicase complexes that are embedded in the membrane. And you're basically passing an electric current through the membrane. And so while the current's being passed through the membrane, double-stranded DNA fragments are basically passed to the helicase. So the helicase unwinds the double-stranded DNA and passes one strand through the nanopore. And now, depending on which nucleotide in particular is kind of being fed through the nanopore at any given time, there's basically some kind of measurable disruption to the current that's being passed through the membrane. And because each nucleotide kind of yields a unique disruption of the current, you can essentially read off a DNA sequence by looking at the pattern of electrical disruption in the membrane. And so here's a hypothetical example of a single-stranded piece of DNA being fed through nanopore protein. And so here the idea is that as you're feeding the sequence through the nanopore protein and measuring electrical current through the membrane, as we feed the sequence through the nanopore and we get different bases passing by, you'll see different changes in current until finally you finish feeding the strand through the nanopore, and you can use basically an equivalent of a base color to look at these patterns of electrical disruptions and basically call bases. And so the last point I'll make is that nanopore sequencing, at least compared to Illumina, for example, is extremely portable. And so here's an example of nanopore sequencing being used on the International Space Station.